Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. Today, I'm delighted to be joined by Graziella Piras, who is a Director of Marketing Bioprocess Segment at 908 Devices. And she is presenting this week at CHI's Bioprocessing Summit on, and I always have to take a deep breath when I read out these titles for presentations, Optimized Cell Culture Medium at your fingertips with at-line spent media analysis. And so today, Chats with Chaudhry is all about the challenges around carrying out at-line amino acid analysis. And you may not guess this, but it's 8 o'clock in the morning Eastern time. And Graziella's kindly got up, got ready, and is sitting in a hotel room, but we should got virtual screen, so you can't tell that, to talk to me with this presentation. Graziella, good morning. How are you?
I am wonderful. Good morning. How are you?
Well, I'm very, very good. I'm very grateful for you getting up so early to do this presentation with me first thing before you get to the show as well. So thank you very much for taking time to talk to me this morning.
Sure, it's my great pleasure to be here and talk a little bit about what we do at 908 and being able to come to a conference in person, right? It's been a long time, so this is very exciting. We're doing all of this with all the safety and measures. We're all being very careful, but very exciting to be able to talk to people in person and being able to talk about what 908 Devices has to offer. So wonderful. Fantastic. It's sunny here in Boston, so all good.
Fantastic. Well, it's a bit cloudy here in Surrey, I have to say, in the UK, and I wish I was there, but actually we're still not allowed to travel to the US. We're still not allowed in, so I have to do this virtually rather than live, but I hope you have a fantastic live show. So before we start talking about at-line amino acid analysis and the challenges around that, can you give viewers who aren't aware a little bit of background on 908 Devices and your own background as well?
Sure, yes, absolutely. Well, I've been in the bioprocessing field for about over 15 years. I joined 908 Devices in April of this year, very exciting. And before that, I really spent my time at Thermo Fisher Scientific in the bioproduction division there, specifically working on solutions for cell culture and cell line development, being in R&D, also in the field, traveling all around the world, and also more recently in marketing. But this is about 908 Devices today. And I joined 908 Devices in April. Very exciting. And basically 908 Devices, our mission is really to democratize mass spectroscopy and make it available at the point of need. Very interesting. 908 Devices has developed sort of benchtop and handheld mass spectroscopy, and they're being used in many different fields. We're going to talk about bioprocessing, but we have quite an amazing device also that is being used by law enforcement and military to detect, dangerous things or drugs. But yeah, in bioprocessing, we have a laptop, a benchtop basically device that we're going to talk about today.
Fantastic. All right. So first question, really basic for a layman like me. What is at-line amino acid analysis? When is it used and why is it important?
All right. So in the field of bioprocessing, to basically create and produce biological drugs that are produced in cells, in living cells, you have to feed those cells with all kinds of nutrients so that they're happy and producing a lot of your protein of interest. For example, monoclonal antibodies, which are used for many different therapies. So how do you feed your cells? You feed them with amino acids, vitamins, sugars, salts, everything to make them happy. But you have to make sure that you're giving them the right amount of nutrients at the right time, all of that. So, and amino acids are central, it's a central nutrient to make those cells very happy. So as you develop your process, you have to know how much, and how much to give them and when. And so this this is why it's so important. It influences not only the growth of the cells, but how well they're going to produce your biologics of interest and how well it's produced as well.
Right. Okay. So what are the main application areas for this type of at-line amino acid analysis?
Right. So it is used in bioprocessing for developing biological drug, namely protein-based, but also in gene therapy, for example, to produce viral vectors for the treatment of genetic diseases, and also in cell therapy, which is, one of the top more recent ways to treat some form of cancers. And there the cell is the therapy, so it's even more important to make sure you're feeding the cells properly so that they're ready to go back and fight that cancer.
Right. Okay. And obviously, there must be some challenges research face. So what are the main challenges then researchers with my dentures in this morning, researchers face when carrying out this analysis?
Right. So, all right. So we know we have to measure those amino acids and vitamins and other things. The traditional way to measure amino acids and vitamins is through HPLC, liquid chromatography, sometimes even mass spectroscopy. But those methods require a very, skilled person, someone who's, an analytical scientist. They take time to develop. You need that skill set, and they take time. Typically, in a process development lab, you have to take your samples. So take the samples from your bioreactor, and then, prepare it, freeze it, send it. If you're lucky, you have a lab within your organization where, a core facility where you can do this analysis, or you have to send it out to a third party. So you can count the days, right? Already there. So before your data comes back, many days have passed, sometimes even weeks. Well, by that time, your bioreactor run is done, and you can't, see if anything went wrong or what is happening, and then you have to wait to set up your next round of experiments. So that's the main challenge, is that amino acid analysis is not trivial. It takes skills, time, and it's costly as well.
Right. So you mentioned Rebel at the very beginning of this conversation. So what is Rebel? Why did you develop it, and what makes it unique?
Right. So the why is really important here, and hopefully you can see where I'm going with this. But because of this main challenge of the analysis being long and costly, etc., at 908 Devices, we thought that we needed to develop, having this mission of providing mass spectroscopy and democratizing it at the point of need.
Right.
We thought this is the perfect, application, and we can really help process scientists get results much faster. The Rebel basically is a very easy to use. I can use it. I'm a biologist. I'm not an analytical chemist. Tool to analyze amino acids, vitamins, and a few other bioamines. So instead of doing everything I said before, what you do is you take 10 microliters from your bioreactor, load it on the Rebel, and after 7 minutes, you know what's in it. You know where the amino acids are, the vitamins, and a few bioamines. So instead of days and weeks, we're talking about 10 minutes.
Right.
And as we know, speed is so important in our field. We just, if you think about, what we just lived through in the past year, bioprocessing is about quality and efficacy, but also speed. And being able to bring a product to market fast is so important. And so we need more and more of these tools that allows analysis of key process parameter and components at the point of need. And the Rebel does exactly that. It's easy to use, small volumes, and it gives you data right away.
Right. And can it be used, in existing PAT workflows as well?
Right. So PAT, process analytics technology, is basically a mechanism developed by the FDA, the regulatory agency, to develop, analyze, and control a manufacturing process, to being able to produce consistently safe and efficient drugs, basically, and it is used by most biopharma and biotech companies. It relies on very frequent at-line or online analysis of key process parameters that may affect what's called the key critical quality attributes of the drug.
Right.
And to do that, you need those tools that are at the point of need, online, at-line, in-line, and so the Rebel fits. As a matter of fact, we have users who are developing PAT process using the Rebel in CAR-T, for example, where it's so important. And again, why is it appropriate? Because of what I mentioned before. It's actually a small footprint, compared to an HPLC or a traditional mass spec. It's the size of a big, maybe like a computer, a benchtop computer. I'm trying to find a desktop computer. It is fast, it's easy to use, you don't need a lot of training, very small volumes of samples, that is also very important. And the data you get is is reproducible and accurate. So it has all the features that you would want, not only in a process development setting, but also in PAT.
Right. And I know that certain industries are moving away from the use of serum in cell culture medium. So how can the Rebel deliver data insight to assist with development and optimization of serum-free media formulations?
Yeah, that is very true. I mean, the trend has been going on for a while, especially in monoclonal antibody production. Barely anyone will use serum. But in these newer applications like viral vectors for gene therapy, or in the CAR-T, these are newer modalities, and sometimes serum is still being used. And serum carries, although cells really like to grow in serum, it comes from, it's a natural product. There's variability, there's sourcing instabilities, and so and risk of adventitious agent contamination. So basically, want to get away from serum, but to replace the serum, you have to basically know what your cells need, how to nurture your cells, and so maybe in analyzing the conditions that are perfect for your cells to achieve their full potential. So the Rebel is ideal for that, because as you grow your cells, you don't want to just take one sample once in once in a while, but you want to take samples throughout your time course. And with the Rebel, because it's so fast and easy, you can do that. You could be measuring, your culture several times a day, which is really very infrequently done right now, because of the, the burden of taking those samples, sending them to the lab, and et cetera, et cetera.
Sure. It's funny listening to you speak. I hadn't really equated, feeding cells like having a diet, and you're basically changing the diet by taking the serum away and having to find a substitute, a meat substitute, almost like saying you become vegetarian overnight, isn't it? So and you have to find out how you can maintain your levels even though you've changed your diet completely. So that's really interesting. So
exactly.
So the other thing is that there are advantages. What advantages are to be gained with frequent at-line spent media analysis when groups seek to accelerate the development work and feed the pipeline? Because obviously everyone's trying to speed things up, aren't they? So how does it help from that area?
Yeah. So being able to gain information more frequently means you're going to get better insight into your process. meaning you're going to be able to understand it better and basically design a process that is more optimized and ideal for your cells. You know, it's all about feeding the cells the right amount, but at the right time as well. So in a typical production run for example, a monoclonal antibody in a fed batch process, your cells are seeded, you know, and they're fed throughout about a 2-week time course. You have to know, you know, you have to feed your cells during this 2 weeks. Well, how much and what, you know, is key. And being able to measure during the time course and maybe even several times a day. Knowledge is power, right? So by knowing more about your process, you can, you know, refine it, fine tune it. And so that's where the RUBL comes in. It's not just taking a sample, you know, once every day, if you're lucky, but you could even do it several times a day. And without depleting your bioreactor, because every time you take a sample, you're taking volume out. But here, you know, it's 10 microliter, about 10 microliters. So you're not depleting your sample, your bioreactor volume.
Brilliant. And so looking ahead, what future applications and workflows can you see REBL being used for?
Right. So I think there are different aspects here. I mentioned, you know, of course, gene therapy with the development of viral vectors, CAR-T right now. So CAR-T is the new therapy, chimeric receptors that are being developed for mostly blood cancers, but of course those are being developed. Similar, you know, therapies for solid tumors. But, you know, we've seen sort of a resurgence of microbial-based, you know, therapies with, for example, mRNA vaccine. The starting point is a plasmid. A plasmid is produced in a bacteria. So we need to refine fermentation processes, get away maybe from undefined broths that are used for fermentation as well, going into chemically defined. And so the REBL is, again, has a great application there. So new modalities, but also we are seeing a completely different industry using cell culture. And I was just attending a conference this week on cultured meat. So there are great efforts to overcome the challenges with meat production in the world, contributing maybe to, you know, pollution. And cultured meat needs the exact same tools. And so we see a lot of interest from that parallel other industry as well. So the sky's the limit, I guess. Anytime you want to have a control process using cell culture, the REBL fits right in.
Brilliant. Okay, so I know the answer to this, but where can people get more information about the REBL if they want to know more?
All right. So I will be giving a presentation on the REBL today and the applications and some examples from our users and how they're using the REBL in some of the applications I've mentioned. That is today at the Bioprocessing Summit in Boston at 3 o'clock this afternoon. We also have a booth in the conference, in the show, booth 604. And also our website, we have a wonderful website where people can see some videos and examples and application notes. Or you can contact us. We're always happy to talk to anyone.
Brilliant, Graziella. Well, that's a very, very thorough and fantastic remembering so early in the morning the stand number as well, although I think you have to find it as well, don't you? That's why you remembered it. So that's really good for sharing that. Thank you very much. Well, look, I appreciate taking time doing this so early in the morning. I mean, I have to say, viewers, if you're watching this, Graziella does look fantastic for 8 o'clock in the morning, you have to say. So well done for making the effort and doing that. Brilliant. Thank you very much. And if you want to know more about the REBL, you can check out Graziella's presentation today. If you this will be shown before the presentation, so you can, if you're at attending Bioprocessing Summit, whether it's virtual or live, you can still check out the presentation. It'll be at 3 o'clock Eastern time, and the link to that will be above the video, as will the link to register for the event if you haven't registered yet, as will the link to the booth be, so you can find out more about the booth, plus the link to the website. So everything you need to know about Rebel, all the links will be above the video. But as Graziella quite rightly said, if you've got any questions for her based on what she said today as well, you can leave some comments and questions below the video as well. I'm sure she'll only be too happy to answer. So Graziella, thank you very, very much for talking to me this morning. It's been lovely to see you, and good luck with your talk this afternoon. I hope it goes really well and lots of people attend as well. So good luck with that.
Thank you so much. Bye-bye.
Bye-bye. And thank you, viewers, for watching. As I said, if you're at Bioprocessing Summit in Boston this week, lucky you. I wish I was there. Make sure you check out the 908 Devices booth. Check out Graziella's presentation. And if you're not there, check out the event virtually. And if you are not there virtually, check out the website and the links above. So until next time, as always, stay well, stay safe. Bye-bye.